1944, groundbreaking experiment by Oswald Avery, Colin MacLeod and Maclean McCarthy. Researchers at the Rockefeller Institute for Medical Research demonstrated that DNA is the molecule responsible for the inheritance of traits in living organisms. Their discovery launched intense worldwide investigation into this complex molecule, but even decades later, DNA remained fundamentally undecipherable. Although scientists knew that the order of adenine, thymine, guanine and cytosine, nucleotides in instance of DNA, somehow determined the characteristics of organisms, they were not able to resolve the sequence of these nucleotides in particular pieces of DNA. As a result, scientists could not establish the relationship between gene sequence and their consequences. This situation changed in the mid-1960s when the combined work of Francis Crick, Sidney Brenner, Marshall Nirenberg, Philip Leder, and Heinrich Mattei revealed that individual amino acids are coded by 3 bases of DNA, a biochemical phenomenon later known as the genetic code. Knowing the relationship between DNA and amino acids changed everything and allowed scientists to see the connection between genes and the basic building blocks for all organisms. A decade later, Walter Gilbert and Alan Maxwell, as well as a separate team of scientists led by Frederick Sanger, independently developed techniques to read the nucleotide sequence of DNA molecules. Researchers then began sequencing genes in a variety of organisms, hoping to understand the relationship between genetic codes and their effect. The long-term goal for many of these researchers was to identify specific patterns of DNA sequences that predispose humans to major diseases. These sequences could be parts of genes or several genes. However, the process of both sequencing genes and identifying their locations on chromosomes was manual and laborious, and in the 1970s, fully analyzing the genome of even a teeny microbe or virus seemed a formidable challenge for the community. In 1977, Sanger and his colleagues successfully sequenced the first full DNA genome, the 5,375 nucleotides long sequence of the 5X174 bacteriophage. As significant a milestone as this work was, sequencing the 3.2 billion base pairs of human genome still seemed nearly unthinkable. In 1983, however, researchers at the California Institute of Technology, funded by Applied Biosystems, used fluorescent text and computer technologies to develop a technique to automate Sanger's DNA sequencing technique. This advance made a dream of sequencing the entire human genome achievable, and a scientific race had begun. One group of researchers, later known as the International Human Genome Sequencing Consortium, won funding from the U.S. Department of Energy and the National Institute of Health in 1990 to initiate the Human Genome Project, intending to sequence the entire human genome by 2005. In 1998, Celera Genomics, a private company in Rockville, Maryland, launched a parallel effort. This competition between the publicly funded and the private efforts led to a tie at the finish line in 2000, when the two groups announced completed drafts of human genome sequence at a televised White House press conference attended by both U.S. President Bill Clinton and UK Prime Minister Tony Blair. By 2003, when the Human Genome Project was completed, the researchers had successfully sequenced nearly the entire human genome with 99.99% accuracy. To this day, many millions of bases that were considered non-coding or junk DNA still await sequencing, and there is a debate about the significance of these remaining non-decoded sequences. Alongside the high-profile quest to sequence the human genome, Many scientists also set out to decode the genomes of rice, fruit flies, E. coli, chicken, mice, and hundreds of other organisms. The enormous amount of data this research has produced has given rise to the new and growing field of genomics. The quest to analyze and interpret genetic data to better understand genetic variation between individuals and different species, develop new treatments for major diseases, and understand human biology. In this spotlight, we invite you to explore the history and present the outlook of this revolutionary field.